Hello, everyone. Welcome back to Global IMMUNOTALKs. Hello, David. And it's great to see you all here again. And as you all know, Global IMMUNOTALKs are and basically started several years ago during COVID by Carla Rotlin and Elina Zoniga. And since then, we had several amazing talks and amazing speakers, and we now have also streaming it live on YouTube. And you're welcome to continue watch the talks also at later time points. And we have been hosting several amazing speakers over the years. And today we have David Veesler. Hello, David, and thanks for joining us today. David is a professor in the Department of Biochemistry at the University of Washington. He is also an HHMI investigator. He has an extensive background in structural biology and viral immunology. And he's particularly focused on understanding the viral structure and the host interaction to develop better therapeutic strategies. And his recent works have been actually seminal findings to understand the molecular mechanism of coronavirus and developing vaccines. And David's work has been crucial in advancing our understanding of this, what actually started Global IMMUNOTALKs, one of the good things that came out of him. And we want to make him, which makes him actually an ideal speaker to our series to discuss what the title of his talk will be, how these viruses can still continue and surprise us. So thank you, David, for joining us today. And I have a surprise question for you. Can you describe the turning point in your career that defined your current research focus?
When I started my lab, I decided to start studying coronaviruses, which I had not been studying before. And we were able to pioneer, the field of entry, how coronavirus enter into cell at the molecular level by actually visualizing these events. That was over a decade ago. And that really allowed us to be very fortunate to team up with so many great scientists, literally tens of collaborators all across the globe, which I'm very grateful for. So many of these collaborations came in response to the COVID-19 pandemic and allowed us all together to develop vaccines, countermeasures against coronaviruses, and try to understand better, to be better prepared for the next time.
Yeah, it's actually one of the good things that came out of this pandemic was the collaborations and the opportunities to do such real-time amazing science. And we're looking forward to hearing your talk. Please, the stage is yours.
Well, thank you very much for the very kind introduction, and thank you for inviting me. I'm going to share my slides. All right, can you see them well?
Yeah, we can see them.
The title of my talk is Can Coronaviruses Keep Surprising Us? And a lot of the talk will be on viral evolution, but I will be connecting these concepts to vaccine development in the spirit of a global IMMUNOTALK. And before starting and sharing with you some of the most recent data from the lab, I thought I will share a couple slides on a question I often ask myself and sometimes discuss with friends in the field. The question is, do we think that Sarbecoviruses are likely to spill over and perhaps to be responsible for another pandemic? So Sarbecoviruses, as most people know, is the subgroup or subgenus of coronaviruses that comprises SARS-CoV-2, SARS-CoV-1, and all these related viruses. And I thought that to answer this question, there are at least two facets to consider. The first one is, well, how diverse are these viruses? And as we all know, the spike glycoprotein mediates entry into cells. And I'm highlighting here the receptor binding domain, or RBD, which is responsible for attachment to the host cell receptor and plays a key role in determining the ability of a virus to invade a new host. So over the past 5 years, we have been classifying Sarbecoviruses not on the whole genome, not even on the spike, but only based on the RBD, which again is the key domain in the context of entry into cell and as well in the context of immunity. So these viruses can be classified in at least 4 clades. The SARS-CoV-2 clade, which we call clade 1b, which is known to use ACE2, which was responsible for the COVID-19 pandemic. The SARS-CoV-1 clade, which we call clade 1a, also utilizing ACE2 and responsible for the SARS epidemic about 20 years ago. Then there's a third clade of viruses found in Southeast Asia, which is a lot less well understood. And there are multiple deletions in the receptor binding domain. We don't know which receptor they use. We don't even know if they are of concern or not. And the fourth clade, which we call clade 3, is found in bats in Africa and Europe. And the receptor was not known for these viruses, and we didn't really know what to make of them either when we started considering this work. So the work I'm going to be showing on this slide was led by 2 graduate students in my lab, Sam Zepeda and Jimin Lee, who worked closely in collaboration with Tyler Starr at the University of Utah and Jesse Bloom at the Hutch. So what we were interested in understanding is how viruses from that divergent clade can enter cells. And what we're able to show is that multiple members, and here I'm showing one of them, can enter cells using a variety of ACE2 orthologs from bats that are shown here on the x-axis. On the y-axis, we are looking at pseudovirus infectivity. So the greater on the y-axis, the more entry we see. So that showed that this clade of Sarbecoviruses is also using ACE2. And what we discovered is that many of them are poised for using human ACE2. They are 1 or 2 mutations away from using human ACE2 efficiently. And actually, one of them is already, without any modifications, able to do so. So this told us that there's really a wide diversity of Sarbecoviruses out there. Many of them can use ACE2, at least 3 of the 4 clades that we know of. That also suggested that ACE2 binding is an ancestral trait among Sarbecoviruses. So now we really understand that there is a large diversity. We don't appreciate the full extent of that diversity. But the second facet to consider if we want to evaluate the risk of spillover and wide-scale spread will be what will happen on the host side. So Min, who is a senior scientist in the lab, and Katie worked together to show throughout the pandemic that neutralizing antibody titers shown on the y-axis are eroded by the constant emergence of new variants of SARS-CoV-2. You can see here when we go towards more divergent variant from the ancestral Wuhan virus, we have a massive decrease in neutralizing activity. This is samples from humans that were infected and vaccinated multiple times. And what's really striking is that the same samples do not show such a drop in binding titers against the spike. We have a strong reduction of neutralizing activity, and binding titers are almost unchanged, about 2x fold change reduction against these highly divergent spikes. So that really suggests that indeed these antigenic changes are selected and emerging to evade neutralizing antibodies, which appear to account for a very small fraction of the total spike binding antibodies in the plasma of infected and vaccinated individuals. What's also interesting is that looking at another cohort back in 2022, we realized that binding titers against SARS-CoV-2 were maintained against SARS-CoV-1 for individuals that were not exposed to SARS-CoV-1. We had a reduction of about 1.5 log. But what we were interested in finding is that would we detect antibodies, cross-reactive antibodies? These are binding titers here. And indeed, although they were reduced against a highly mismatched virus such as SARS-CoV-1, we could clearly find these antibodies in the plasma of these individuals. Then what about effector functions? If we have binding antibodies, perhaps we have Fc-mediated effector functions, and we actually evaluated antibody-dependent cellular cytotoxicity with the samples shown on the top row. And what we saw is that indeed they were able to mediate approximately comparable ADCC against cells in vitro expressing each of these variants. And that's in line with the fact that binding titers don't seem to be affected too much. And that's a good thing, because it is highly likely that Fc-mediated effector functions are a contributor, perhaps a main contributor, to retain protection against severe disease in case of infection with SARS-CoV-2 for individuals who have been vaccinated. And a last illustration of the importance of effector function, that's an example from a paper from Mike Diamond's lab at WashU. And I really like this example because he studied the impact of a potent RBD-directed antibody against SARS-CoV-2 in the context of therapeutic administration, meaning multiple days, well, it might have been 1 day after infection. So it is clear that the antibody provides full protection against weight loss. However, if you mutate the Fc fragment to remove Fc effector function, all of a sudden you have a marked reduction of the ability of this antibody to protect in a therapeutic setting, really highlighting that effector functions are important not only against mismatched viruses, but again, also against matched viruses, in that case, in the context of administration post-exposure. So the fact that most of us will have been exposed to vaccines and most likely infection with SARS-CoV-2 suggests that there is widespread Sarbecovirus antibodies in the plasma, or the ability to make them in the memory B cell compartment of individuals. And that suggests to me that a pandemic of the scale we have seen with the Sarbecovirus is not very likely. Obviously, it does not deny the fact that there is still infection and transmission to an appreciable scale. People are also suffering from long COVID, and all of that is very important. But I think it's unlikely that we can experience a Sarbecovirus pandemic of the scale we have seen just because immunity exists in the human population. So one of the questions that my lab has been interested in understanding over the past few years is trying to find out which viruses should we be most worried about. And the reason for that is there are so many viruses discovered in wildlife. Here I'm showing an example with a pie chart showing sequences discovered in bats. There are viruses in many other animals, but in that example, you can see more than a dozen viral families for which sequences were found in bats. A lot of them were coronaviruses in that specific example. And that's quite daunting. We can't study them all in depth, and we need to be able to prioritize which ones to work on to develop countermeasures and vaccines and be ready for next pandemics. So one of the things we are trying to do is to bridge the gap between the discovery of a large number of sequences in wildlife, here the example is coronaviruses, and our very limited understanding of the likelihood of each of these viruses to cross the species barrier and infect humans. So clearly, for a virus to be successful, many factors need to be aligned. The presence of a host receptor, an activating protease provided by the host as well. And once a virus enters cells, it needs to be able to antagonize host innate immunity and hijack cellular machineries to replicate. So these are really a lot of parameters. But for the context, in this talk, I will be focusing on the first two steps that really lead to viral entry. Is there a proper receptor, and is the virus able to enter into cell, to try again to bridge this gap between the large number of viruses out there, and we probably know only a small fraction of them, and our very limited understanding of the likelihood to cross the species barrier. So on this slide, I'm showing a summary of the 9 currently known human-infecting coronaviruses. What I would like you to take away from that slide is the color code that I'm showing is that these viruses are using a finite number of receptors. I'm showing 9 viruses, but only 4 receptors, and actually only 3 of them are used again and again. And what's happening is that if I were to show all the coronaviruses found in wildlife along with the ones in humans and the receptors they are using, I would not be adding much on that slide in terms of receptors. I would just be adding many more viruses using the same receptors again and again. What's common to all these receptors is that they are transmembrane proteases for which the catalytic activity is not required for coronavirus entry, meaning that the docking platform used as a receptor is not the protease that is providing the cleavage events following entry. So my talk will be focusing on a subgroup of coronaviruses called Merbecoviruses, or the Merbecovirus subgenus. This is the subgroup of viruses related to MERS-CoV, which was discovered in 2012 in Saudi Arabia, and that has been infecting humans at a small scale, but that has become almost endemic in the Arabian Peninsula ever since. And MERS-CoV is using DPP-4, the receptor. It has a very high fatality rate of about 35%. And most transmission are occurring from camels, which are widely infected, to humans, and some human-to-human transmission have been described as well. So one of the questions we started asking is, well, how diverse are these viruses? How many more viruses are there, and how worried should we be besides MERS-CoV, which is already a main concern? So we decided to classify these viruses based on RBD sequences, just like I showed you for Sarbecoviruses. And we were able to delineate 6 clades shown here in different colors. With at the bottom, the MERS-CoV clade using DPP-4, the receptor, and the HKU4 clade, which is the most closely related, also using the DPP-4 receptor, led to the assumption by the field for more than a decade that DPP-4 will be a universal receptor for Merbecoviruses. Then the other clades are even more divergent at the RBD level, and the receptors are not known. I would like to point out how divergent these viruses are from each other. To put that in context of Sarbecoviruses, the most divergent Sarbecoviruses will show about 70% amino acid sequence identity in their RBDs, whereas here we are finding viruses with as little as 30% amino acid sequence identity. We all know how challenging it is to develop a vaccine that will elicit high titers of neutralizing antibodies against all Sarbecoviruses. And here we are looking at twice as much genetic divergence. And so that really emphasizes the grand challenge ahead of us to be able to develop broadly protective countermeasures against these viruses. So to try to understand better, again, if these viruses will be of concern, we decided to study the most divergent clade shown at the top. And with 2 viruses in particular, I apologize in advance, the names are not easy to pronounce. One of them is called NeoCoV. The other one is called PDF-2180. I will call it PDF moving forward. These viruses were found in bats in 2 different African countries. And what's really interesting to me is the fact that they are among the most divergent from MERS-CoV at the RBD amino acid sequence level, but they are classified as the most closely related to MERS-CoV because of their high whole genome nucleotide sequence identity. However, again, when we care about entry into cells and immunity, the RBD is really the business part. And so here we have an incongruence between these 2 ways of classifying or looking at viruses. And I think the advantages of RBD classification will become obvious in the coming slides. So this work is a fantastic collaboration with Hugh and Hien at the Taikang Center for Life and Medical Sciences. We decided to team up a few years ago because we discovered we were interested in similar aspects and we're working on overlapping questions. So we joined forces, and the discovery of the receptor for NeoCoV and PDF viruses was actually entirely serendipitous, based on the concept that coronaviruses appear to be reusing the same receptors again and again. It was an obvious idea to test if the known receptors will work for these viruses. So here you can see that MERS-CoV and HKU4 enter cell using DPP-4, as I mentioned, but not ACE2 or APN. And so what Huon's lab found was that ACE2 could serve as a receptor for Neo and PDF-2180, which was really highly surprising because it was completely unexpected that ACE2 would be a receptor for any Merbecoviruses. And so to try to confirm and extend these results, screening of a large panel of bat ACE2 orthologs revealed that actually a large number of these ACE2s were mediating entry, as shown in the heat map. The darker the red, the more entry for each of these ACE2 bad orthologs for these two viruses. And the human ACE2 is shown here. It's like actually very pale, although it is a receptor. It's a very weak receptor compared to many of these bad receptors. What was even more surprising is to realize that almost all non-BAT mammalian ACE2 orthologs tested could actually mediate entry into cells, as shown here on this heat map. And that really revealed the completely unexpected finding that ACE2 was a receptor, was used again by a group of coronaviruses with Merbecoviruses, and that the species tropism of these two viruses was almost unprecedented based on how diverse the ACE2 orthologs that can be used are shown on these plots. Finally, the ability to use the human ACE2 receptor, although weakly, was of concern in the possible ability of these viruses to spill over in the future. So to understand entry into cells by these viruses and the molecular basis of receptor recognition, we determine cryo-EM structures of the RBD of these viruses and the whole spike, and the RBD is bound to ACE2. I'm showing you just one example here with the RBD of NeoCoV bound to ACE2. And what's interesting is that the region involved in binding is the tip of the RBD and involves multiple glycans from the bad ACE2, which are important for entry, as if you mutate the glycans, you completely abrogate entry for one of the two viruses and markedly reduce it for the other virus. This ACE2 recognition mode is entirely different from the DPP4 recognition mode from MERS-CoV, which is using the side of what we call the receptor binding motif in the RBD here, and besides entirely different chemical surfaces, the architecture of the RBD with elongated loops will be incompatible with ACE2 binding. So sequence analysis revealed that NeoCoV and PDF-2180, when comparing the genome with the one of MERS-CoV, underwent a recombination event in the S1 subunit, swapping the RBD, and that most likely brought the DPP4 specificity to Merbecoviruses using DPP4, such as MERS-CoV and HKV4. So now we realize that ACE2 could be utilized by one clade of Merbecoviruses. We started asking the question, is that an exception among Merbecoviruses? Because there are so many more viruses for which receptor utilization remains elusive. So we decided to look at the MOW clade shown here because they are just as divergent from MERS-CoV than the ERI-CoV NeoCoV clade is, but they are also about 30% amino acid sequence identity to that clade. So they are very divergent from everything else. And they have a unique feature found only in these two viruses that are forming that clade, in that there are two insertions in the RBD, in the region presumed to be the receptor binding region, that sets them apart from all other Merbecoviruses. So screening orthologs from various BATs and non-BATs ACE2s, we realized that these two viruses could also bind, as shown in green, and enter cells expressing a very limited number of ACE2 orthologs, which really is different from what I was showing you from NeoCoV, and a very small subset of non-BAT mammalian ACE2s were also serving as a receptor. Here, receptor usage, as I just mentioned, is very narrow and excludes human ACE2. And using cryo-EM, we determine structures of the RBD of each of these viruses bound to ACE2. And the clade-specific insertion I showed on the previous slide are actually responsible for receptor binding. So it's a different binding mode than the one I showed. And there's actually a key residue at the interface, which is glycosylated not in the receptors, but in all non-receptor ACE2s. And the presence of this glycan is a steric obstacle for receptor recognition, explaining why some receptors are actually functional and some are not for this subgroup of viruses. So at this point, we started realizing that ACE2 usage is a lot more widespread among Merbecoviruses than what we had previously appreciated. And what was absolutely amazing is to map the binding sites of each of these two RBDs from the NeoClade and the MOW clade on the ACE2 dimer shown here in the membrane. As you can see, the binding sites are entirely different. They are more than 50 angstrom away from each other. And that suggested to us that these two clades evolved independently to use ACE2 as a receptor. So we're super excited by these discoveries because that was totally unexpected. And then we started wondering, now that ACE2 is just as widely spread as DPP4 in terms of the number of clades using ACE2 or DPP4 in the Merbecovirus subgenus, what is happening with the other viruses? And that's again an example of coronaviruses showing to us that the answer was just in front of us all along, and we just did not look carefully enough. The same receptors are being reused again and again. And it turns out that for the HKU5 clade, which was discovered, the first member HKU5 was discovered in 2006, almost 20 years ago, in P. abramus bats in Hong Kong. It turns out that P. abramus ACE2 is a receptor for that clade. Had we tried 20 years ago, we would have realized it right away. It was not obvious to us that there was this pattern of reusing the same receptors. But here again, ACE2 is a receptor with a small number of non-bats, mammalian ACE2s that were also functional, but not human ACE2. So HKU5 is clearly able to use ACE2 as a receptor. Again, human ACE2 is not, suggesting that HKU5 will not be able to use human ACE2 as is, although I will get back to that point. To understand receptor engagement, we determine cryo-EM structures, and that work led by Young-Jung Park, a fantastic staff scientist in the lab. And based on this information, we were able to map key regions in the receptor shown in dark green that were mediating the ability or not to use a given receptor. And we showed by mutagenesis that mutating each of these three residues, or the three together, abrogated binding and pseudovirus entry into cells. When we map the binding sites of each of these viruses, we realize that HKU5 yet again utilizes a different ACE2 surface and a different set of residues, suggesting that Merbecoviruses evolved ACE2 utilization at least 3 times independently. So if we integrate the structural data and the mutagenesis I showed, we're able to obtain a blueprint of where a given virus recognizes, in this case, HKU5, the receptor with a footprint. And we are also able to pinpoint what we call key determinants of receptor species tropism. They are shown in navy here, and these are the residues that if they are suboptimal, they will hinder receptor utilization and vice versa. It turns out that the human ACE2 receptor has suboptimal residues for HKU5 at each of these positions, suggesting to us that human ACE2 will not be a receptor, and which is compatible with what I just showed you, that it does not mediate pseudovirus entry and no binding could be detected. However, we carried out the experiments using authentic virus, which is sometimes more sensitive because it's not a single round assay, it's a multi-round replication assay. And what we saw is that in the presence of exogenous trypsin, human CACO2 cell endogenously expressing ACE2 or overexpressing ACE2 led to detectable expression shown as red puncta or replication of HKU5. And that's actually confirming findings that were made a long time ago by Vinit Menacheri when he was working in Ralph Berrick's lab at UNC. But it turns out that this replication with human ACE2 is very, very weak compared to the native bat P. abramus ACE2 receptor. However, the fact that the replication was even detectable led us to ask, is HKU5 actually poised for human emergence? So to answer this question, we decided to use recent breakthrough in machine learning, especially in computational structure prediction and protein design. And something I haven't told you yet is that each time we determine a new structure in the lab, we always try to predict it to see how well predictions are matching our data. And in the case of Merbecoviruses, everything I showed so far was actually not working at all with AlphaFold 3, but I'm sure it's going to improve very rapidly. However, HKU5 was really an outlier because AlphaFold 3 was clearly able to understand what the interface looked like, which we were super excited about. So I'm showing you here our cryo-EM structure in cyan and the AlphaFold 3 prediction in red, and they are almost identical, at least for the interface region, which is what we cared about. So again, we found that very exciting because we thought that we can use these tools to accelerate our work and answer these questions much more rapidly. So we then asked if AlphaFold 3 thought that ACE2 would be a receptor, and AlphaFold 3 led to a bunch of nonsensical predictions, which is what it will tell. This is a sign that it actually doesn't think that the two proteins are interacting, which is great because in that system, not only it could predict the interaction with P. abramus, but it could also predict the fact that we could not detect binding to ACE2 because it is so weak. Because we had identified the key receptor species determinants, we then decided to mutate the 3 key positions in human ACE2 to match the P. abramus host ACE2 sequence that allowed to see entry, and asked if AlphaFold would realize that these mutations were sufficient to enable entry. And indeed, introducing the 3 mutations allowed to predict the structure accurately of the complex. Now, to try to understand if HKU5 is actually poised, and that would be a change in the virus and not in the receptor. So we use protein MPnN developed in the Baker lab here at the University of Washington that allows to design an optimal sequence given a structure. And here we are just trying to identify if a small number of mutations at the interface with these key receptor species determinants would be sufficient for HKU5 to use human ACE2. We were able to find a single mutation that was actually predicted. This is a predicted structure, not an actual experimental structure, but a tyrosine to lysine mutation predicted to form a salt bridge that would enhance human ACE2 binding. And we decided to test that experimentally using purified recombinant proteins or single-round non-replicating pseudoviruses. And what Jack in our lab found is that this single point mutant that we identified enabled detectable, albeit weak, human ACE2 binding, whereas the wild-type sequence did not. And Jimin found that pseudovirus entry was detectable for the single point mutant, but not for the wild-type sequence, only in the presence of trypsin, indicating that it's a very weak receptor still. But that retention of the P. abramus ACE2 receptor was present, suggesting that this is a mutation that could evolve naturally in the host and that would allow ACE2 utilization. So I would like to point out that, again, all these experiments were not made with authentic viruses, so these are safe experiments. There's no replicating virus, and these are purified proteins on the left-hand side and pseudovirus on the right-hand side. But the ultimate validation of our results came from an independent publication out of Zhang Liqi's lab, who discovered directly in bats an HKU5 strain that was unknown and that can already utilize human ACE2. The mutations are different from the ones we identified computationally, but that really show that indeed these viruses are poised for human emergence. So the last clade, in the interest of time, I will not describe it too much, but we found that it also utilizes ACE2, and it's not a new mode, but it's a mode identical to the HKU5 clade, although the details of the interaction are different. So at the end of the day, it turns out that ACE2 utilization is much more widely spread in Merbecoviruses than anticipated, and DPP4 is clearly not supporting entry of any of the other clades besides the ones that have been known for a long time, MERS-CoV and HKU4. We can actually map the binding sites of all 5 groups of coronaviruses using ACE2, besides the 3 Merbecovirus clades. There's SARS-CoV-2 and SARS-CoV-1, but the 2 are identical binding modes, and L63, which is a human endemic coronaviruses. And they all utilize different structures and bind different sets of residues, indicating that there is a possible 5 or more independent acquisition of ACE2 utilization among Merbecoviruses. So one of the questions that came to our mind is, why are coronaviruses doing that? Why do they keep reusing the same few receptors? And although I don't think we have a final answer yet, I will try to touch on that and share my thoughts of where we are at the moment. To best answer this question, I will share a movie that we had actually put together back in 2019 with Janet Iwasa at University of Utah, showing MERS-CoV entry into cells. And what I really want to show is the mechanical aspects in which the RBDs have to open up for receptor binding to occur, then the activating protease will come. But what's important is that once the receptor binds, the RBD is stuck, and then the conformational changes of the fusion machinery can take place. So we actually describe these conformational changes in 2018, 2019, and the fact that the receptor blocks the RBDs in the open state, preventing closing and therefore releasing the constraints on the metastable fusion machinery, resembles what we call a molecular ratchet. Once you start going in one direction, you just can't go back. And I think this is one of the key aspects that all these molecules have in common, is that to trigger the conformational changes leading to fusion, a receptor needs to be able to engage the receptor binding domain when it is open. Usually it occurs with a surface that is occluded in the closed state, and therefore, once it is bound, closing of the spike or of that RBD is impossible, and then you drive the direction of the conformational changes in one direction. And so to try to understand, again, better, what is important, our hypothesis was really the molecular ratchet. We asked, can we replace parts of the receptor? And the short answer is actually you can replace every single part of ACE2, literally every single part, transmembrane domain, cytoplasmic domain, and it can still serve as the receptor. The only requirement is that for the region recognized by the virus to be able, of course, to bind to the receptor binding domain, but also to have this molecular ratchet mechanism fulfillment. So replacing the peptidase domain to which all these viruses are binding by a mini binder, which we described many years ago, which also recognizes a surface that is only accessible in the open state, but not in the closed state of the RBDs, is working as a receptor. And we can even shorten or lengthen the distance between the mini binder and the membrane. And what's interesting is to realize that there is really an optimal distance. If it's too short, you drop entry by multiple logs. If it's too long, you also drop entry. So it appears that part of the reason, there are probably more reasons, but part of the reason these receptors are being selected is that they are fulfilling the molecular ratchets mechanism requirements and some geometric constraints that will be compatible with triggering these conformational changes. With that, I would like to conclude and thank all the wonderful members of my lab that are shown here, and our fantastic collaborators, Hugh and Hien at the Taikang Center for Life and Medical Sciences, Tyler Starr at the University of Utah, and David Ecorti at UMABS, and all the funding sources. I would like to thank you for your attention. I'll be happy to take any questions.
Thank you very much, David. It was very interesting and, as expected, very enlightening. Now we are going, as you know, there are no talks, there are no questions that you can ask live. The questions are all going to be online, and you can direct them and ask them specifically on YouTube, on the channel there. You have the information you can find here, scan it, or just go to YouTube to Global IMMUNOTALKS. You can also ask them on Twitter and other avenues that we now have account. Also, I want to remind you that next week we are going to have Min Peng talking in the Global IMMUNOTALKS, and we are all looking forward to seeing you on our next Global IMMUNOTALKS. Thank you very much, David.
Thank you so much for having me.